everyone, today we'll be looking at Biology Unit 1.1 Part 1. In this lesson, we'll be learning about cell theory and what a prokaryote is. So, the cell theory is one of the most fundamental principles of biology. It states that all organisms are composed of cells, all cells come from pre-existing cells, and the cell is the smallest living organizational unit. So, in the cell theory, it's stated that all cells come from pre-existing cells. This is also known as biogenesis. Henry Carlton Bastian found about biogenesis, meaning the generation of life form from non-living materials. But Thomas Henry Huxley modified the meaning to our current one. Until the 1850s, spontaneous generation was accepted as the origin of small organisms like maggots. In other words, maggots could suddenly be formed from anything, even a grain of sand. However, it was disproved in 1859 by Louis Pasteur, when he boiled beef broth in 2 flasks, both with glass goosenecks. So this is what a glass gooseneck looked like. So, when he broke one of the goosenecks, the broth was exposed to the air and microorganisms began to grow in. This was how he proved that spontaneous generation was not valid. Louis Pasteur also showed that boiling and cooling wine and milk killed all microorganisms living inside it. That is why the process of pasteurization was named after him. So inside a cell is the organelles. They all have their own specific roles, and these are just the most common and basic features. So, the plasma membrane. This is the boundary of the cell. However, in plant cells, there is also a cell wall to support its structure. The cytoplasm is the fluid or interior of the cell. This can also be called the cytosol. This is where all the work is done. DNA, also known as deoxyribonucleic acid, carries hereditary information and controls the cell's activities. The DNA is often stored in the nucleus. This is what a nucleus looks like. Ribosomes are sites of protein synthesis. Synthesis means to make, so ribosomes are where proteins are made. So the classification of living things. All living things are classified into many sections. However, we'll only be looking at domains and kingdoms today. There are 3 domains: bacteria, archaea, and eukarya. So, bacteria and archaea are separate domains because they are prokaryotes. However, they are completely different from each other, so they are in the same domain. And we'll go through what a prokaryote is later on. There are 5 kingdoms: bacteria, archaea, protista, plantae, fungi, and animalia. So prokaryotes. Prokaryotes are all unicellular bacteria and archaea. Prokaryote cells lack membrane-bound organelles, and their cytoplasm contains scattered ribosomes that are involved in the synthesis or creation of protein. The chromosomal DNA is attached to the plasma membrane by a region of the chromosome called the origin. So this is a chromosome cell, or this is what a chromosome looks like, and this is the origin. So many prokaryotic cells also contain small rings of double-stranded DNA called plasmids. The pili and flagellum are hair-like projectiles often around the cells. They help move the cell and they help the cell to move and transfer DNA between other organisms. Prokaryote cells are much smaller than eukaryotic cells, and therefore they have a larger surface area relative to their volume, which allows the cell to take in and release materials efficiently and replicate quickly. So bacteria. Bacteria are prokaryotes. Fossil evidence dates back 3.5 billion years ago, which confirms that they were the first living organisms on Earth. Bacteria has cell walls made of peptidoglycan, and many secrete an outside capsule for protection. Bacteria is very diverse and can survive in a large range of habitats and conditions. However, they are most commonly found in moderate temperatures that are moist and low in salt, where sunlight or organic compounds are plentiful. They can be photosynthetic, chemosynthetic, or heterotrophic. This will be covered in Chapter 2, about autotrophs and heterotrophs. Photosynthetic and chemosynthetic autotrophs are organisms that create organic substances using inorganic material. Photosynthetic autotrophs requires sunlight and the process of photosynthesis, whilst chemosynthetic autotrophs uses the chemicals from chemical-rich habitats like hydrothermal vents deep in the ocean. In these hydrothermal vents, the bacteria would use compounds such as sulfides or ferrous ions instead of the sun and photosynthesis. Heterotrophs are consumers. They rely on eating other organisms to obtain their energy. So bacteria, it can also be gram-positive or gram-negative. So gram-positive and gram-negative bacteria. The cell walls of a prokaryote contains murine, also known as peptidoglycan, which is a giant molecule consisting of sugars linked by amino acid. In most bacteria, the murine forms a cell wall outside the plasma membrane. There is this purple stain called crystal violet, which is used to identify gram-negative and gram-positive bacteria. Gram-positive bacteria have a thick layer of murine that absorbs and holds the stain, giving them the purple color. The gram-negative bacteria can't absorb this stain, which makes them pink. So this is gram-positive and gram-negative. You can see the difference in color. So examples of gram-negative bacterium is the cyanobacterium, which can produce toxins capable of killing fish and other aquatic life and cause illness to humans. So you see this green patch. This is cyanobacteria. Archaea. Prokaryotes in the domain Archaea include extremophils, which means they live in extreme conditions. This can include very high or low temperatures, pH levels, which is acidity, salt levels, and oxygen levels. However, some Archaea live in less extreme environments, like the open sea. Archaea have a different plasma membrane structure that enables them to tolerate extreme conditions. And the unique habitats of Archaea were not recognized for a long time because the extreme conditions were difficult to find and maintain in laboratories. Although bacteria and Archaea look extremely similar, they are two completely different organisms. Archaea have their unique membranes. Like other organisms, their membrane is composed mainly of lipids. Lipids are organic compounds including fats and oils composed mainly of carbon, hydrogen, and oxygen. Lipids have proportionately less oxygen than carbohydrates. These cell membranes need to be fluid to respond to external deformations and damage. The lipids in eukaryotic cell membranes, which is like animals and plants and fungi, have stability and selective permeability, but only in narrow ranges of temperature. In the Archean membranes, the structure is formed to remain fluid and permeable over a wide range of temperatures, from freezing cold to boiling hot. This is how they tolerate these extreme conditions. So the difference between bacteria and archaea. So archaea have a different type of lipid structure in the plasma membrane, which enables them to live in these extreme conditions. And the cell wall in bacteria contains murine, but the cell walls in archaea do not. Both have diverse metabolic systems, but methanogenesis, where methane is produced, is only unique to archaea. And so this is the end of part 1. In the next part, so this is the end of part 1, chapter 1.1, biology. And in the next part, which will probably be uploaded next week, we'll be going through eukaryotes and what they are and the different types of microscopes. And we'll finish unit 1.1. Thank you for listening. Don't forget to subscribe and give a thumbs up. Yeah. And if you have any questions below, just type it into the comments and I'll try my best to answer it. Yeah. Thanks.